Hi, my name is André Marques. I'm a group leader at the Max Planck Institute for Plant Breeding Research in the Department of Chromosome Biology. In my group, we're interested in understanding how holocentric chromosomes that can have hundreds of centromeric units, can influence and affect genome organization, karyotype evolution, and also meiosis. And today, I'll be telling you about a very nice story where we found that repeat-based holocentromeres can influence the genome architecture and also the karyotype evolution in plants with holocentric chromosomes.
In contrast to monocentric chromosomes, as you can see here, a holocentric chromosome lacks a primary constriction, and centromeres can extend from almost one telomere to the other telomere. We first starting by comparing the chromatin organization between a monocentric and a holocentric genome at interface. In contrast to the cluster distribution of centromeres in a monocentric genome, holocentric genomes with repeat-based holocentromeres are actually characterized by having hundreds to thousands of centromeric units which are specifically associated with a specific repeat, the so-called TIBA repeat, which is also specifically enriched for CNH3, the centromeric histone protein. Although they are distributed genome-wide and dispersed at interface, they come together and they form the so-called holocentromeres at metaphase chromosomes. We now compare the chromosome structure between a monocentric and a holocentric chromosome at the metaphase state. Monocentric chromosomes are characterized by having the primary constriction where the centromere is located, where both sister chromatids are glued to each other. However, holocentric chromosomes lack the primary constrictions and both sister chromatids are glued to each other along the whole extent of the chromosome. Basically, in a monocentric chromosomes, we have a very nice and distinct compartmentalization of genomic features. For instance, the centromere is localized in a single region and size restricted. Also around the centromere, we have the so-called pericentromeric regions, where we have the main concentrations of repeats and also heterochromatic marks. So what we label here in orange should be silenced or at least mostly silenced regions. In contrast, genes tend to accumulate more at the chromosome arms, where we highlight here in light blue, and is also highly euchromatic and highly transcribed. However, in contrast, holocentric chromosomes with repeat-based holocentromeres lack this organization. Euchromatin and heterochromatin are basically uniform distributed along the whole chromosome. We now highlight the main difference between a monocentric and a holocentric chromosomes. And as you can see, holocentric chromosomes lack the so-called compartmentalization in different subdomains as in a monocentric case. We now compare chromosomes at anaphase state and how this can impact at chromatin contacts. In monocentric chromosomes, normally the microtubules attach at a single region of the chromosomes, as you can see here, at the centromere, which normally causes the so-called V-shaped migration of sister chromatids. This also causes that regions that are distantly located in a single chromosome may also interact to each other, the so-called long-range interchromosomal interactions. This will also cause long-range interactions between different chromosomes, the so-called long-range interchromosomal interactions or trans-interactions. However, in holocentric chromosomes, microtubules attach along the whole extent of the chromosome, and this will cause the parallel migration of sister chromatids. Another main difference here is because they lack the so-called V-shaped migration, also chromatin contacts will be impacted, and we lack these long-range intrachromosomal interactions. This will also impact in a reduced frequency of long-range interchromosomal interactions. I hope you enjoyed our study, and if you want to know more about it, please read our paper that's now out in Cell. Thank you.